Eryops is from 295 million years ago from the Texas red beds up in Archer County. What's the results of our decalcification on Eryops? Liberated osteocytes. Osteocytes are the little bone cells that live in bone. They make bone. They dissolve bone. Did you know that your skeleton is recycled every 12 to 15 years? Completely recycled by these little guys that live in there. Yeah, they're cute. They're fun. We're going to show you osteocytes from these cells. We're going to show you tendons, not only liberated, but in the bone. We're going to show you fully clotted vessels, and we're going to show you clot canopies. What's a clot canopy? Well, you'll have to stay awake, and we'll show you. These are cells from Eryops, 295 million years old. The preservation gets better as we go back in time. There are more branching on these little thread feet. These are the little extensions that come off the osteocytes, and they touch each other. All these cells are touching each other. North, south, east, and west, every direction, all these cells are touching each other or talking to each other at the same time. It's almost like the neurons in your brain. This is in your bone. So here's Eryops osteocytes. Look at this field of cells in collagen and look at all the connection points between them. The preservation gets better the deeper we go in the fossil record. There it is. This is a liberated tendon. This is for tensile strength in the bone. And I'm going to show you pictures of bones that look like pin cushions with these things sticking out. But here one is liberated. It's under polarized light and it's showing strain inside. This is an extruded fiber. It's like a plastic rod. And you can see some of the particulate matter in here that's part of it. But it's showing you the stress. In polarized light, if you take a water bottle and cut off a piece of that and put it under a polarized light microscope, you'd see all kinds of colors in it. Why? It's under stress. It's extruded and frozen in that condition. That's the way these tendons are. They were created and frozen in that condition. So we're finding tendons liberated. Well, I told you I'd show you a pincushion, didn't I? So you're looking at a piece of Eryops bone that we decalcified partially. These things I'm finding are decalcifying 10 times faster than Triceratops and Nanotyrannus. I have to literally pull them out and wash them to stop the decay because it happens so quickly. But I want you to note all these vessels still in the canals. That's a vessel. This is a tendon. This is a tendon. All of the other ones are vessels. And look at how dark it is inside. It's fully clotted. And the vessel wall is still there. We didn't see this in Triceratops or Nanotyrannus. So vessels are intact in this bone. There's one tendon just sticking straight up out of the bone. And notice it's clear. So it has no pigment or anything. Here's another shot. You can see it from a little bit different angle. But what we're picking up now are these canopies. And here's a vessel that obviously wound through a canal. We erased the canal, right? We took the bone out. And so there's the vessel, fully clotted. These canopies are up at the top of the vessel near the surface of the bone where capillaries would form. And so even the capillaries clotted, and we have those showing up as a canopy like a tree canopy. I don't know about you, but I'm astounded by this stuff. These are impossible pictures. That's what I call these. Impossible. They can't be there. Not supposed to be there. And they're there. Here's a beautiful picture of a clot. I'm going to show you a video of one in a second where we'll focus in and out, and you can see the canopy right up there at the top. But all these sticking up out of the bone, fully clotted. So that was Eryops. Let's talk about Cacops. I showed you that one. He's a lot shorter. He's only about 5 inches or less in length. This is 230 million years old from Richard Spur, Oklahoma. What did we find? Liberated cells. Now we're having liberated nerves. You saw tendons in Eryops. Here's nerves in Cacops. Anybody know what a lipid is? Raise your hand if you know what a lipid is. What is it?
Fat cell.
Louder. Fat. It's fat. It's like oil. It's like olive oil. Imagine pouring olive oil into the Cretaceous and walking away for 10 years, 20 years, 100 years, 1,000 years. This is 230. It has lipids in it. I'm going to show you movies of lipids and pictures of lipids, clotted vessels, and chondrocytes. These are like osteocytes, but they're in cartilage. So we have preserved cartilage with cells in them. Again, beautiful cells, wonderful preservation. You might even be able to make out internal organelles inside these cells. Look at the network. All the collagen now has gone away because these things sit on a collagen carpet that gets filled in with bone mineral. But look at all the connections in here. I've never seen this in Triceratops or Nanotyrannus or any of Dr. Schweitzer's work with T-Rex, the sum of it, but not like this. 230 million years old, Richard Spur, Oklahoma now. Oh, am I going backwards? I am so sleepy, guys. I haven't slept in 3 days, so.
Stand up.
Okay. I don't mind being short, I tell you, I really don't. This is a liberated nerve from Cacops. Now, you're going to have to watch this morning's lecture because one of the things we showed in our paper that was published, remember this is unpublished, was the crosshatch pattern in the nerve. The collagen connective tissue that surrounds the nerve fascicles. These are the nerve fibers where the electricity travels. The connective tissue that surrounds this is weaved across all of it, all the way up and down the nerve in a 45-degree angle crosshatch pattern. And you can begin to see a little bit of it in here and up in here. You can see that crosshatch pattern. But this is exactly like the nerves that we found in Triceratops. I have a colleague who works on some of my specimens sometimes, and he sent me this picture, and he called it unknown fiber. I said, no, it's not an unknown fiber. Look at the crosshatch. That's a nerve from Cacops. Beautiful picture. I promised to show you lipids. I have worked a lot with lipids. I thin-sectioned probably 12 bombardier beetles and made maybe 300 slides, and they're full of lipids. The bombardier beetle, as you know, has to produce a reaction instantaneously. It needs a lot of energy, so it has large lipid bodies in its abdomen. So I know what they look like. I've stained them before. They react to osmium. And here are 2 lipids being squeezed out of the nerve that have been stained in osmium. Now I can do this because I use a really long objective that touches the slide, and as I focus, it puts pressure. And I'm going to show you some video of me doing that, and you're going to watch the nerve swirling all around. But I can actually squeeze the lipids out by putting pressure on the nerve, and they just pop out. We saw this in our class this week. I was showing the students lipids from these nerves. Here is a thin section now of a clot in a vessel. So here's the canal. The clot didn't go into the bone. All of these pits here, these are pits where the osteocytes lived. So this morning when I said that contamination can't be true because you'd have to have an osteocyte crawl through the dirt, find the dinosaur bone, crawl in, and set up in the exact pattern. How does an osteocyte know I need to turn left or right? Come on. Contamination is not an issue. We know that we're looking at these tissues because we're seeing the diagnostic structures that define them. Here's the, I just froze my brain. Cartilage, thank you. Here's the cartilage, and you can see actually cells dividing in some of this. You can see crenated outlines of cells. So some cartilage has been published, but not a whole lot, and definitely none this old.
Would you repeat that? Those cells are dividing?
This cell right here is dividing. That's a mitotic plate. Now, this is not the best picture of a mitotic plate. Dr. Schweitzer in her group actually published late 2019 a beautiful picture of actual chromosomes lined up along that mitotic plate. It wasn't mitosis, it was apoptosis that puts the chromosomes in that configuration, but nevertheless she imaged chromosomes. Yeah, in cartilage. So we're not the first ones to do that. So we have corroboration of nerves, clots, and cells between this whole time period. So we are hoping to dig at the Permian sites in about a month. We need your encouragement and we need your support because all the structures you've seen have come out of these bones that have been out of the ground for decades. I can't imagine what we're going to find when we dig fresh material.